Hello, I'm Melissa Spencer, and I'm a professor of neurology at UCLA. And my lab is interested in using genetic engineering technologies to develop therapies for neuromuscular disorders. So neuromuscular disorders are diseases that impact either skeletal muscle or nerve. And my lab is primarily interested in studying skeletal muscle. And we know that there are at least 500 different genetic causes that have been defined for these disorders. And the advantage of studying a monogenic disorder is that you know the cause, and then you can use gene type editing or gene replacement to develop very effective cures. And so we are focusing on three main areas. The first is to develop these genetic engineering technologies, either CRISPR or gene replacement, and a lot of the CRISPR work is being done with the PAL lab. We're looking at platforms to deliver these therapies, so either nanoparticles with the Meng lab or using viral vectors, primarily AAV. And we're also interested in looking at immune responses to these viral vectors, primarily through single cell sequencing of peripheral blood cells, but also standard immunological approaches. And a newer project is to use PET imaging in collaboration with the Ikuton lab to track these viral vectors throughout the body. And I'll mention this a little bit more later. So why do we care about the immune response to AAV? We know that a single dose of AAV will reach its intended target. In our case, it's muscle. So we can give a dose of it. It will transduce the muscles of the body. It won't be 100% efficient, but we can have pretty good efficiency with these high doses. However, AAV is limited to single dosing. And so if we were to try and give a second dose to a patient, what we would see is that there would be an immediate immune response. The viral vector and the gene would be cleared before it would reach the muscles, and we wouldn't be able to have any efficacy at all. And so we're very interested in studying what these immune responses are so that we can block them. And also, we know that about 70% of people in the community have been exposed to wild type AAV. So the AAV that's used for gene therapy is recombinant AAV, but it's very similar to wild type. And so 70% of people already have pre-existing immunity to AAV. And so we'd like to understand what the responses are to be able to block them, to allow us to redose, and also to allow us to dose patients with pre-existing immunity to develop highly effective therapies. And so we know a little bit of the immune response that results. We know that there are both innate and adaptive immune responses, but very little has been done to really comprehensively profile what cells and what messages are being turned on that can be targeted. So that's one aspect of the lab. So as far as potential rotation projects, one option would be to assess these immune responses to AAV vectors. This is already ongoing in my lab, but a student could potentially take on one particular aspect or design a new experiment to inhibit the immune system or a component of the immune system, and then to do single cell sequencing to evaluate further that effect. The second would be to design new CRISPR approaches using new platforms and technologies as they arise, or to target a disease we're not currently studying. And the third is more exploratory, would be to use PET imaging to track AAV biodistribution and persistence. And as I mentioned, this is being done with Dr. Ikoton in the Department of Pharmacology. And this would be a very green project, but very potentially exciting. So those are the three main areas where a student could potentially do a rotation. So my lab is located in the Neuroscience Research Building, which is very close to the NBI, shown here on the map, this blue dot. And here's what the building looks like. My contact info is on this slide, and you're welcome to reach out to me with any questions you may have. Thank you.